Hello everyone, welcome you all. So today's lecture we will be discussing about microsporogenesis. Microsporogenesis, how it takes place in the anther, right? So let us start the lecture. So here is a diagram in front of you. So we were talking about how the microsporogenesis takes place. So all of you know how typical flower is having how many walls? 4 walls a typical flower has. That is calyx, corolla, androecium and the gynoecium, isn't it? Androecium and the gynoecium. So we were discussing about this male part, male reproductive organ, and its unit is called what? Stamen. Its unit is stamen. So you know that each word of a flower is a leaf. It is a modified leaf. So stamen is what? It is a microsporophyll. Microsporophyll. So fill term here which we are using is representing what? A leaf. So this is a modified leaf. We used to call it microsporophyll because here we will see the formation of microspores. Alright, so a typical stamen is equivalent to a microsporophyll. Now let's go inside. Stamen is having a bilobed anther. Generally the anther is bilobed and there is a filament. So this is anther and this is the filament. Okay, so now let's see what is present inside the anther. Let us take the section of it. Let's take the transverse section here. Now this is what is depicting inside the anther. Okay, so what you will see inside a typical anther, you will see there are 4 layers. What are 4 layers here? From the outside we are entering inside. The first layer here is the epidermis. First layer here is the epidermis, right? The outermost layer. Second layer is the endothecium. There are middle layers around 2 to 3 in number, and the fourth layer here is the tapetum. So a typical anther is having how many layers? 4 layers are present over there. Okay, outer one epidermis, lower one tapetum. Epidermis, endothecium, middle layers and the tapetum. In this way you have to remember. So what I said, a typical anther is having how many lobes? 2 lobes. So this is a bilobed anther. Anther can be unilobed. In some families like Malvaceae it has 1 lobe only, but here we were discussing about a general structure of anther which is bilobed. Now if you see the section, you can see there are 2 lobes, right? This is 1 lobe, this is the second lobe. Now in each lobe there are 2 theca. 1, 2. Here also 1, 2. So a typical anther is bilobed, dithecous, tetrasporangiate, and it is tetragonal. Remember these 4 terms. Bilobed, having 2 lobes. When you cut the section, you can see each lobe has how many theca? 2 theca or 2 pollen sacs. These all are what? These are the pollen sacs in which the pollens are formed. And at every corner there is microsporangia. So in total how many microsporangia are there? In total there are 4 microsporangia. 1, 2, 3, 4. So typical anther is tetrasporangiate. There are 4 microsporangia. So anther is equivalent to microsporangia. It is equivalent to microsporangia. So remember these terms. Microsporophyll is stamen. Microsporophyll is stamen. Anther is equivalent to microsporangia, and your pollen grain is equivalent to male gametophyte. Male gametophyte. So inside the gametophyte, gamete is formed. Gametophyte is a body which form what? a gamete. So I hope the layers are clear. Outermost epidermis and endothecium, then we have what, middle layers, and the innermost is the tapetum. Right, all the things are clear till here. Now inside the tapetum there is what, this is what, this is a sporogenous tissue. This is a sporogenous tissue, which is, each cell is having the potential to behave as microspore mother cell. This is a mother cell which is going to produce what? It's going to produce microspores by meiosis. We will see that how these microspores are produced. Okay, so 4 layers. Inside there is what? Sporogenous tissue. Every cell of a sporogenous tissue is what? It is the potential microspore mother cell. Let's proceed ahead. So how, when the from the starting only there are we cannot see the 4 layers here, but in the starting we have epidermis layer, and this is the outermost epidermis, and inside that cells are present which are having the potential to divide. Now what happens, there is a differentiation of one cell at every corner. This cell is called archisporial cell. Okay, this is archisporial cell. Now this archisporial cell divide to form 2 cells. Outer one is parietal cell and the inner one is sporogenous cell. Outer one is parietal and inner one is sporogenous. Okay, so archisporial cell divide periclinally, and the outer one is called parietal and the inner one is sporogenous cell. Now this parietal cell is going to form the wall layers. It is going to form the endothecium, the middle layers, and the tapetum. So these are the wall layers. And the inner one, the primary sporogenous cell, this is going to produce what? This is going to produce sporogenous tissue. So this is what happening in the young anther. Now when the anther matures, you can easily see there are 4 layers, right? The outer one is epidermis, then endothecium, there are 2 to 3 middle layers, and the tapetum. Inside there are numerous sporogenous cells which is having the potential to behave as microspore mother cell. And you can easily see a stomium here. This is what? This is a line of dehiscence. From here only the anther dehisces. This is called line of dehiscence. Right, now the part, this is the anther and this is the filament, right? This is the filament. This filament is extending upward. Okay, and here this is what? The line of attachment where you will see the vascular bundle. Okay, this is connective. Here we will see the vascular bundles are present. Alright, now moving ahead. Now the microspores which are formed inside anther, the typical microspore is tetrahedral or isobilateral. These are the most common microspore formation. Beside these two, you can also see decussate, linear or T-shaped type of microspore formation. So decussate you will see in magnolia, linear in asclepias, T-shaped in aristolochia. So these are not that much common. The common one is tetrahedral or isobilateral. In tetrahedral, 3 cells are visible to you easily. One is at the back side. So total 4 are formed. Now let us see how the microspore is formed from the sporogenous cell which behaves as a microspore mother cell. Right? So whatever is written here, I hope it is clear. Microsporangium, it is surrounded by 4 layers. There are 4 layers. We have seen epidermis, endothecium, middle layer, and the tapetum. The starting 3, these are going to help in the protection. They have a protective function and they help in the dehiscence. Especially this endothecium. Endothecium has fibrous thickening. Which helps in the dehiscence of the anther. The innermost layer, that is the tapetum. This is very important. Many questions are asked from this layer. Tapetum nourishes the developing microspores. This is a nourishing layer. Okay, and this layer also produce Ubis bodies. Ubis bodies, these are coated with the sporopollenin, this we will talk about, which causes the thickening of the exine. Exine is thick. If you see the structure of a pollen grain, there you will see the outermost layer is present, which is called exine, and it is having thickening, which is made up of sporopollenin. And this tapetum layer also produce oily material called pollen kit in entomophilous plant, in which the pollination is by insect. Pollen kit is a sticky material which helps in the pollen kit is a sticky material which helps the pollen grain to stick on the insect body. Okay, so pollen kit is present. So this pollen kit is produced by the tapetum layer only. So the main function of the tapetum is to provide the nourishment to the developing microspore. And the tapetum can be uninucleate, or it can be multinucleate. So in a single cell there can be more than one nucleus. It can be multinucleate, right? Okay, proceeding ahead now. When the anther is young, we have seen a group of compactly arranged sporogenous tissue occupy the center of each microsporidium. Now if you talk about each sporogenous cell, suppose this is a sporogenous cell which is having the potential to behave as microspore mother cell. Now what it do? It undergo meiosis, right? It undergo meiosis 1, and then meiosis 2. Ultimately, how many cells are formed? Total 4 cells are formed, right? So first of all, karyokinesis occurs, and then cytokinesis. Karyokinesis is what? Nuclear division. Cytokinesis is cytoplasm division. So microspore mother cell, this is diploid in nature. Remember this, microspore mother cell it is not a microspore yet. It is a microspore mother cell. It undergo meiosis 1 and then meiosis 2. And we have how many cells? From one microspore mother cell, we have 4 microspore formation. 4 microspores are formed from one microspore mother cell. It is also called pollen mother cell. Is that clear? So if I would say, if in a typical anther 100 microspore mother cells are there. How many? 100 microspore mother cells. So how many microspores are formed from 100 microspore mother cells? You will say how many? 100 into 4, 400. Is that clear? Now if the question is like this, if in a microsporidium there are 50 microspore mother cell in one microsporidium, how many microsporidia are there? 4. From one cell, how many microspores are formed? 4. 200 into 4, 800. So in this way, how many microspores are formed? 800 microspores are formed, right? So these types of question you will get in your exam. Proceeding ahead, now as the anther is maturing and these microspores which are formed from the microspore mother cell, they also dissociate from one another and they develop into a pollen grain. Now if you see the structure of a pollen grain is having how many layers? Pollen grain has 2 layers. Outer one exine and the inner one is intine. Exine has a thickening of sporopollenin, and this intine layer, it is made up of pectin and cellulose. So it is pectocellulosic in nature. It is rich in pectin and cellulose. Of course, there is a plasma membrane inside. There is a plasma membrane. Now, inside there is a large vegetative cell vegetative cell. Okay, the nucleus here is irregular. These are the vacuoles. And a spindle shape generative cell is present. Spindle shape generative cell. This is generative cell. So this is the structure of a pollen grain. Okay. So when the anther dehisced pollen grains are released, these pollen grains are called the male gametophyte because here the male gamete is formed. A typical pollen grain, how many cells? Vegetative cell and generative cell. Generative cell is small spindle shape. Okay, it is spindle shape, and this cell undergo mitosis to form two male gametes. These are male gametes. Okay, so as I told you, generative cell is haploid, because pollen grain, it is a haploid structure. Every cell is haploid here, whether it is generative cell or a vegetative cell, all the cells are haploid. Now if a haploid cell is forming a haploid cell, what it will do? It will do equational division, which is mitosis. Is that clear? Because microspore mother cell is diploid in nature. Microspore mother cell, it is diploid in nature. It has to form the microspore. Microspores are haploid. So microspore mother cell will do what? Meiosis division. Meiosis is reductional division. Right? So this pollen grain is a haploid structure. It is uninucleate. Haploid, uninucleate, and minute spore, which is generally spherical. There are two layers, intine and the exine. Outer layer exine has thickening of sporopollen, and this sporopollen is very, very important layer. It is most resistant organic material. No enzyme can degrade it, and it is absent at the prominent aperture called germ pores. So at some places you can see this layer is not present. This sporopollen is not present. There is a aperture which is called a germ pore. From here only the pollen tube arises. Right? So at some places you can found a germ pores are present. Okay, so this is a typical structure of a pollen grain. Intine, exine, these are the two layers here. Then a large cell, which is also called tube cell. It is vegetative cell, also called tube cell, has a nucleus which is called tube nucleus. It is irregular nucleus. Spindle shaped generative cell is floating inside a vegetative cell. This is a generative cell. Now important to note here is pollen grains are shed as two-celled stage. Generally, one vegetative cell, one generative cell. So in 60% of angiosperm this is happening. Now what about 40%? In 40% the vegetative cell divide to form two male gametes. So already the pollen grain have three cells now. So one vegetative cell and two male gamete. So how many cells? 3 cells. So in 60% the pollen grains are shed at two-cell stage. Generally it is happening, but in some cases you can also see that the generative cell divide mitotically to form two male gamete before the shedding. Alright. Vegetative cell is larger cell here. A vegetative cell is a larger cell. It is having a lot of nutrients. Okay, has abundant reserve food material and a very large nucleus. Generative cell is small, as I told you. It is a spindle shape and it is floating in the cytoplasm of the vegetative cell. Clear? Okay. Pollenology is the study of pollen grain. Remember this. Pollenology is the study of pollen grain. Logy is study. Okay. Now once the pollen grain is shed, it has to land on the stigma. Is that clear? What is the motive of the pollen grain? It has to land on the stigma. This is the stigma. Clear? So pollen grains has to land on the stigma. If it is not landing on the stigma in a proper time, they have some viability. They are viable for some period only. If they are shed on that time only, then only they germinate. Otherwise they are waste. They will not germinate. So once the pollen grain are shed, they have to land on the stigma before they lose the viability. Okay. Now viability vary.